Hi everyone, this is Anitha from St. Joseph's College of Arts and Science, Autonomous Cuddalore. So today in this video we are going to see about the topic endoplasmic reticulum. So this is a structure of endoplasmic reticulum, which is a meshwork or network of three components, namely cisternae, vesicles and tubules. So when you speak about endoplasmic reticulum, it is a network of membrane-bound cavities, vesicles and tubules. It is distributed throughout the cytoplasm. And this main role of endoplasmic reticulum is biosynthesis of proteins and lipids. The term endoplasmic reticulum was introduced by Porter in the year 1948. And this endoplasmic reticulum, it will be absent in eggs, embryonic cells, RBC and bacteria. The space inside the endoplasmic reticulum is called as lumen. This is the structure of endoplasmic reticulum. So when you speak about its structure, it contains three components, namely cisternae, vesicles and tubules. So speak about cisternae, they are long flattened unbranched sac-like structures and they are arranged in parallel bundles. Its diameter is of about 40 to 50 microns and they have ribosomes on their surface and they are normally found in the secretory cells. So second component is vesicles. So vesicle, it will be rounded or ovoidal structure and they have the diameter of about 25 to 500 millimicron and they are found aberrance in case of pancreatic cells and they are found at the end of cisternae and then tubules. Third component is said to be tubules. So tubules are branch-like structure and they have diameter of about 50 to 100 millimicrons and they normally occur in non-secretory cells like muscles and they arise from cisternae. So these are all the various components of endoplasmic reticulum like cisternae, vesicles and then tubules. The membrane of endoplasmic reticulum is made up of phospholipids and then proteins. And the main component of endoplasmic reticulum is phospholipids and proteins and it is a fluid mosaic-like structure. Its thickness is of about 50 to 60 angstrom. The membrane of endoplasmic reticulum constitutes more than half of the total membrane of an animal cell. The membrane of endoplasmic reticulum is continuous with that of plasma membrane, Golgi membrane and the nuclear membrane. The lumen of endoplasmic reticulum acts as a passage for intracellular transport. So next we move on to types of endoplasmic reticulum. So endoplasmic reticulum is classified into two types. One is said to be granular and another is said to be agranular. So granular is otherwise called as rough endoplasmic reticulum. Agranular is otherwise called as smooth endoplasmic reticulum. So first we are going to see about granular or rough endoplasmic reticulum. In some endoplasmic reticulum, the spherical granular structure is called as ribosome and it will be attached on the surface. So when it is attached on the surface of endoplasmic reticulum, then it is said to be granular endoplasmic reticulum. The binding site of ribosomes on rough endoplasmic reticulum is called as translocan and it occurs in almost all cells which are actively engaged in protein synthesis such as liver cells, globular cells, pancreatic cells and plasma cells, and it is in the form of flattened sacs. So second type of endoplasmic reticulum is said to be agranular or smooth endoplasmic reticulum. So here the ribosomes are not attached on the membrane of the endoplasmic reticulum. So they are said to be agranular or smooth endoplasmic reticulum. And it is a system of tubules. And it will be mainly present in retinal cells. So it is called as myeloid cells or myeloid bodies. and it is present in the muscle cells, then it is said to be sarcoplasmic reticulum. So next we move on to different types of enzymes present in the endoplasmic reticulum. So these are all following enzymes that is present in the endoplasmic reticulum. First one is NADH cytochrome C reductase, glucose 6 phosphatase, magnesium activated ATPase, nucleotide diphosphatase, uridine, guanosine, inosine diphosphatase to hydrolyze UDP, GDP and then IDP. Synthetase for synthesis of phospholipids, glycolipids, phosphologens, fatty acids and then steroids. So next we move on to functions of endoplasmic reticulum. So there are different functions are performed by the endoplasmic reticulum. So first one is mechanical support, that is it gives support to the cell. So endoplasmic reticulum, it divides the fluid component of a cell into two different components by which it gives mechanical support to the cell. So hence it is said to be cytoskeleton of the cell. And second function is transport. So it acts as a circulatory system involved in import, export, intercellular circulation of various substances. So by this process, proteins, lipids, enzymes, everything is transported with various parts of the cell. So this is the second function performed by the endoplasmic reticulum. Third one is protein synthesis. So main thing regarding endoplasmic reticulum is it contains ribosome. So main function performed by ribosome is it involved in the protein synthesis. So due to the presence of ribosomes, the main function performed by the endoplasmic reticulum is it acts as a protein factories, and amino acids are assembled on ribosome to produce polypeptide chain. Then the ribosome will be attached to the endoplasmic reticulum, are more efficient in protein synthesis than free ribosomes lying in the cytoplasm. So this is third function. Fourth one is formation of microbodies. So microbodies are said to be subcellular organelles, whereas endoplasmic reticulum, mitochondria are said to be organelles, microbodies are said to be subcellular organelles. So they won't be well defined as that of organelles, so they are said to be subcellular organelles. So microbodies are small granular bodies filled with an electron dense granule rich in peroxidase, and they include peroxisomes and glyoxysomes. So endoplasmic reticulum is bud of the microbodies, that is only from the endoplasmic reticulum microbodies are formed. And fifth function is synthesis of cholesterol and then steroid hormone. So endoplasmic reticulum acts as a major site for synthesis of cholesterol, which is a precursor for steroid hormone. So it can be any kind of an hormone. The precursor will be cholesterol. Only when there is cholesterol, the steroid hormones will be synthesized. And in the testis, ovary and then adrenal cortex, the smooth endoplasmic reticulum plays a major role in the synthesis of steroid hormones. And next one is glycosylation. So glycosylation means addition of carbohydrate unit to other cellular molecules. And then it leads to the formation of glycoprotein, mucopolysaccharide, glycolipids and then glycogen. Glycosylation is catalyzed by an enzyme, glycosyl transferase. So next function performed by the endoplasmic reticulum is detoxification. So detoxification means removal of toxic substance is said to be detoxification. Detoxification is an important function performed in case of liver. So in case of endoplasmic reticulum, detoxification involves in biochemical reactions by which harmful materials are converted to harmless substances. So detoxification occurs in endoplasmic reticulum of liver cells. So next function is lipid synthesis. So endoplasmic reticulum synthesizes the triglycerides, phospholipids, and it also acts as a storehouse for lipids. That is, lipid is not only synthesized in case of endoplasmic reticulum, but it also stored in the endoplasmic reticulum. Next function is glycogenolysis. So glycogenolysis means the conversion of glycogen into glucose is called as glycogenolysis. It takes inside the endoplasmic reticulum. The endoplasmic contain an enzyme called as glucose 6 phosphatase, which will be involved in the glycogenolysis. And this enzyme which converts glucose 6 phosphate into glucose, which is transported to the blood. So this glycogenolysis mechanism, it will be takes place in the endoplasmic reticulum. So next function is storage of calcium ions. So endoplasmic reticulum not only act as a perform different function, but it also act as a storage for calcium ions. That is, endoplasmic reticulum stores the calcium ions. When the muscle is stimulated, that is, in case of muscle contraction, the main component involved for the muscle contraction is calcium, which will be stored in the endoplasmic reticulum. So, when the muscle is stimulated, endoplasmic reticulum releases rapidly the large amount of calcium ion into the cytosol, and this brings about muscle contraction.